All right, hello everyone, TA Reese here. So today we are going to go over discussion number 3. So hopefully y'all saw the beginning of this in person in class this week. If so, I'll add the minute mark that you can fast forward to catch up where we left off in class. If you did not make it to class this week, I decided to send out all the recordings to everyone for Unit 1. However, once we get to Unit 2, that will change. So you do need to come in person for Unit 2 materials or you will not receive the recording. I'm currently in the biology office and I just printed off copies of discussion number 4. However, I'll only have 10 extra copies per class. So please print this out. I did edit it and add a lot of extra animations or shapes and stuff. Slides. So please print this off. I will only have 10 extra copies per section. So yeah, awesome. So let's go ahead and get into discussion number 3. And again, this is our last discussion before exam 1. So let's begin. So here is the PowerPoint for discussion number 3, and what we are looking at are hormones and hypothalamic control. So how the endocrine system and the nervous system interact, specifically the central nervous system. That's where the hypothalamus is located. So question number 1, we have 3 patients at an endocrinologist's office, and they all have thyroid disorder. So 3 patients at a hormone doctor, an endocrinologist, and they all have a specific disorder of an endocrine gland, the thyroid. So you can see in these pictures here, the thyroid is located in the neck region, and it is a very common gland to be disordered. So thyroid disorders are very common. And so in order to address the following questions throughout this discussion, we're first going to draw a flow chart illustrating the hypothalamic anterior pituitary pathway. So we're first just going to figure out the basics on how this pathway works, how we send hormones to different regions, and how we release hormones from specific endocrine glands. In this illustration, we're going to include some organs and also hormones involved. So let's begin. So this will be under that first area on that first question on the discussion worksheet. So the first thing that happens involving our hypothalamic anterior pituitary pathway is we have some type of stimuli. So this stimuli can be external or internal. It could be a bee landing on your arm. It could be your stomach being empty and you feeling hungry. There will be some type of stimuli, and that stimuli is going to first arrive at the hypothalamus. So some external or internal stimuli will occur and it will be processed first by the hypothalamus. The hypothalamus will process that stimuli and send a signal to the anterior pituitary. So if you see in this picture I included here, the hypothalamus is this little tiny region of the brain, kind of right there at the middle and the base. And right below it, we have the pituitary gland. So the hypothalamus and the pituitary gland are connected and they work together to really carry out a lot of important functions in our body. So the anterior pituitary will receive that signal from the hypothalamus, and then it will send another signal to a specific endocrine gland. So we have many different endocrine glands in our bodies. Endocrine glands release hormones. The main ones we're going to look at in this class are the thyroid glands. Again, those are found here in the neck and the cervical region. We have the adrenal glands. So the adrenal glands sit on top of the kidneys and they release adrenaline. So epinephrine and norepinephrine, and they also release our stress hormone cortisol. We also have the gonads or the sex glands, so the ovaries in the females and the testes in males. And of course, there are many other endocrine glands in the body, but those are the main ones that we're going to focus on in this course. So that anterior pituitary, the first gland of the endocrine system, also known as the control center of the endocrine system, will send that signal to the specific endocrine gland, and that specific endocrine gland will send that signal to a target organ or tissue. So some target organ or tissue where we will have a response to our original stimuli. So this is that basic pathway. Let's go ahead and add some extra notes here. So one thing we see is that our hypothalamus is considered the integration center one. So the first integration center. So we receive that stimuli and we integrate it in the hypothalamus. When I say integrate, I mean we decide what to do with it. The hypothalamus can release many different signals. And what we'll see here in a second is when I say signals, I mean hormones. But the hypothalamus can release many different signals or hormones. Therefore, we have to integrate that initial stimuli to decide which one of those hormones we want to release. Again, we're going to release that first hormone to the anterior pituitary gland. That is our integration center 2. So that is the second place where we integrate that hormone sent from the hypothalamus. So a hypothalamus is going to send out a specific endocrine or a specific hormone to the anterior pituitary, and the anterior pituitary as well can release many different types of hormones. So it'll integrate the hormone received from the hypothalamus and decide which hormone it wants to release. Again, decide and integrate go along with each other. The anterior pituitary integrates that hormone and then sends it to the endocrine gland, which we consider the final integration center. In this class, we don't see any centers in between the anterior pituitary and endocrine gland. However, in other cases and working with other endocrine glands, there might be a middleman in between the anterior pituitary and the endocrine gland. So we don't consider it IC3, integration center 3. We instead consider it the final integration center. So I've been saying signal for the most part. The hypothalamus releases a signal to the anterior pituitary, which releases a signal to the endocrine gland, which releases a signal to some target tissue. When I say signal, what I really mean is hormones. So let's go ahead and add this now. So our hypothalamus will release hormone 1. And that hormone 1 will be sent to the anterior pituitary, which will release hormone 2. Hormone 2 will be released from the anterior pituitary and travel to our specific endocrine gland, which will release hormone 3. So one thing that's important to know is we consider hormone 1 and hormone 2 the tropic hormones. So we call hormone 1 and 2 the tropic hormones because they have a cascading effect. So if we increase the release of hormone 1, we will increase the release of hormone 2 and increase the release of hormone 3. They have that cascading effect. Whatever happens to one will happen to the next 2. However, hormone 3 works in the opposite way. It works through negative feedback. So it works through a negative feedback loop. And this is how a normally functioning hypothalamic anterior pituitary pathway works. So what this means is that if we increase the release of hormone 3, we're going to decrease the release of hormone 1. If we decrease the release of hormone 3, we'll increase the release of hormone 1. So it acts in this opposing direction, and that's why we call it negative feedback. And it's very, very important. I'm going to give you an example, and we'll get into some deeper examples in just a little bit. But if you're like, why do we use this negative feedback loop? Well, let's talk it through. So let's say we have some type of stimuli and it's a stressor. So we have a stressor stimuli. If we have some type of external or internal stress applied and sent to the hypothalamus, the overall hormone that we're going to want to release is cortisol, and cortisol will be released from the adrenal glands. Cortisol is our stress hormone. It's normally released with adrenaline as well. So whenever we release cortisol, what happens is our heart rate goes up, our pupils dilate, our vessels dilate, we start breathing heavier. We go into that fight or flight state. And that's good whenever we have some type of stressor applied and need to go into that state. However, we don't want to stay in that state forever. That would be very bad for our bodies if we were in a constant fight or flight state. So once we cause the cascading effect to release cortisol, we will produce it, release it, and then we will signal back to the hypothalamus, hey, the adrenal cortex released enough cortisol. We're good now. Let's decrease the release. So if we increase the release of cortisol, we'll go into fight or flight, but we don't want to stay there forever. So we'll signal back through negative feedback to decrease the release of cortisol. And we're going to work through some specific examples to really make this click coming up. But that's just a general idea of what's going on here. All right, so moving on. So now we're going to draw out the specific anterior or the hypothalamic anterior pituitary pathways for these 4 hormones. So if you want to draw on an extra sheet of paper, I would definitely recommend writing this down. So first we have cortisol, then we have thyroid hormone, then we have growth hormone, and then we have our sex hormones. So we're going to draw out that pathway for each, and then we're going to talk about the specific hormones that come from each center. So if you remember, what was our first integration center? It is the hypothalamus. And then the hypothalamus is going to release a hormone to our second integration center, which is the anterior pituitary gland. The anterior pituitary gland will release a hormone and send it to our specific endocrine gland, which for cortisol, as I said earlier, is the adrenal cortex. So that's the general pathway for every single hormone that we release to our body. It always, that stimuli always is, recepted by the brain, by the nervous system through nerves in our body. We receive it in the nervous system. We release a hormone to the endocrine system control center, which is that anterior pituitary. And then from there, we release a hormone to a specific endocrine gland in our body, which will release another hormone to cause some type of response. So I really want y'all to just have it down that we go hypothalamus, anterior pituitary, specific endocrine gland. I just want y'all to get that down, because what's more important to know is which specific hormone we're releasing from each. So this is the very basic pathway for each. The thyroid hormone specific endocrine gland is the thyroid gland. The sex hormone specific endocrine gland is the gonads. So in females, it would be the ovaries, in males, the testes. And then we don't state a specific endocrine gland for the growth hormone because growth hormone can be sent to many different areas of the body. It can be sent to the muscles to stimulate the production of muscle tissue. It can be sent to the skeletal system to stimulate the production of new bone tissue. It can be sent to the integumentary system to stimulate the production of hair. Specifically with growth hormone, we see the growth of chest hair. With growth hormone, we don't go as complex on which specific area we send it to. We're going to consider the anterior pituitary the final region here. So again, you need to just have that down. Now we're going to add the specific hormones released from each, and this is what you really need to focus on. So we have some external stimuli that tells us that we have some type of stressor, and we need to release cortisol, or stress hormone. So what we see is that we receive this stimuli at the hypothalamus, and the hypothalamus will release CRH, cortico-releasing hormone, cortico-releasing hormone. So the hypothalamus will release cortico-releasing hormone and send it down to the anterior pituitary. The anterior pituitary will then release ACTH, adrenocorticotropic hormone, adrenocorticotropic hormone. So the anterior pituitary will release ACTH, and that will be sent to the adrenal cortex, our specific endocrine gland for the release of cortisol, and the adrenal cortex will release cortisol. So this is that pathway and the different hormones that are integrated and released to the different regions that we use to release our final hormone, here specifically cortisol. So quick review, the first 2 hormones released, what do we refer to those as? Those are going to be the tropic hormones. So those are the tropic hormones, because again, they have this cascading effect. If we increase the release of hormone 1, we increase the release of hormone 2 and increase the release of hormone 3. And we're going to work through some specific examples of that coming through, but I just wanted to kind of ask you that, one time before to help you instill this knowledge. So now let's move on to thyroid hormone. So what we see with thyroid hormone is we have some type of stimuli that arrives at the hypothalamus and tells us, hey, we need to release some thyroid hormone. So the hypothalamus will integrate that stimuli and it will release TRH, thyroid releasing hormone. So the hypothalamus will release thyroid releasing hormone to the anterior pituitary gland, and the anterior pituitary gland will release TSH, thyroid stimulating hormone. So the anterior pituitary releases thyroid stimulating hormone, which will stimulate the thyroid gland to release thyroid hormone, which is technically called T3, T4. Dr. Chang normally just refers to it as T4. I don't believe in lecture you go over the difference of T3 and T4. They're just known as thyroid hormone. They both work in pretty much identical mechanisms. So that's the pathway and the specific hormones released for thyroid hormone, which that's going to be the main hormone that we focus on in this class. So next we have growth hormones. So we have some signal or stimuli that is received by the hypothalamus, and the hypothalamus will integrate that stimuli and first release GHRH, growth hormone releasing hormone. So the hypothalamus will release growth hormone releasing hormone and send it to the anterior pituitary gland, which will release GH, growth hormone. So it releases GH, which will travel to many different areas of the body to either stimulate the production of muscle tissue, skeletal tissue, hair, and many other things. The growth hormone has a lot of functions in the body. And lastly, we have our sex hormones. So we have some type of stimuli that is integrated by the hypothalamus, and the hypothalamus will release GnRH, gonadotropin releasing hormone. Gonadotropin releasing hormone. So the hypothalamus releases that to the anterior pituitary, which releases either FSH or LH. So FSH, it's follicle stimulating hormone, and LH is luteinizing hormone. Both are released in males and females, but both have different functions in males and females. Luteinizing hormone has to do with egg and sperm cell production. FSH in females deals with follicle production, follicle stimulation, and has minor functions in males, but is still released in males. So whenever we release FSH and LH from the anterior pituitary, they travel down to the gonads, which again in males is the testes and females is the ovaries. And the gonads release either testosterone or estrogen, our two sex hormones. Common misconception, many people believe that only males have testosterone and no estrogen, and females have only estrogen and no testosterone. However, both males and females have testosterone and estrogen, just in very different levels. So females definitely have testosterone. Females with very defined jawbones and very defined muscles normally have higher levels of testosterone. Males with softer jawbones and cheekbones and kind of softer and harder to grow muscles, they normally have higher levels of estrogen. They exist in both males and females. Testosterone is commonly misused as a steroid because if you take testosterone steroids, you can grow muscle tissue easier. You also can define your cheekbones, your jawbones. However, it does have negative side effects. It does lead to increased growth of chest hair, increased growth of upper lip hair, things like that. Estrogen can also be taken as a supplement. So estrogen can be taken to grow breast tissue. And it can also be taken as a form of birth control. So both can be taken supplementally wise, but it's not recommended for sure to take testosterone. And then estrogen is only recommended to take if you have talked to your doctor and have a plan for birth control. Awesome. So again, hypothalamus, anterior pituitary, and specific endocrine gland, just have that down. What you need to focus on more is for the specific hormone we're trying to release, what's hormone 1, what's hormone 2, and what's hormone 3. So really focus on that. All right, awesome. So let's go ahead and move on. So now we are on to our second set of questions. So we are doing patient data analyzation, which this is really, really cool stuff. what we're doing here is what they do in hospital and clinical settings. So we're analyzing data that we take from patients in the hospital and clinical settings. They would do this through blood draws, analyze the specific hormones in the blood, things like that. So what we're learning today is very interesting stuff. This is stuff that most of the population has no idea how to do or how it's done. And today we're learning that. So be proud of yourself. This is very, very interesting things. So our first patient is named Colin, and what we see is that he has a rare hypersecreting hypothalamic tumor. So it's our job to see if we can compare him to one of these data sets. Now, these data sets are generalized and they are definitely provided in the hospital and clinical settings. They might not say data set A, instead data set A will line up with a specific condition or disorder. So this could line up with hyperthyroidism or Graves' disease or blah, blah, blah. We'll see some specific examples of that coming through, but these charts are provided in hospital settings to do these comparisons. So let's go ahead and start off with doing these comparisons. So the first step here is we're going to draw out our hormonal pathway for thyroid hormone, because again, these people have thyroid disorders. And how we work through this is by first drawing out that hormonal pathway. So who remembers what the first hormone is in our thyroid hormone pathway? That is TRH, so thyroid releasing hormone. That will be released from the hypothalamus and cause the release of thyroid stimulating hormone from the anterior pituitary. Thyroid stimulating hormone will stimulate the release of which hormone? Thyroid hormone from the thyroid gland. So that's that hormonal pathway for thyroid hormone. So now that we've drawn out our pathway, now we're going to draw out some arrows. And these arrows are going to indicate whether we're increasing the production and release of one of these hormones or decreasing the production and release of one of these hormones. So how we start is we look at the condition we're given. Colin has a rare hyper-secreting hypothalamic tumor. So what this tells us is that he is increasing the release of one of the tropic hormones. He is causing this increased cascading effect of increasing release of all of these. So we'll start with TRH. So TRH, we will draw an increase or up arrow next to it. So we're increasing the release of thyroid releasing hormone, which again, that's a tropic hormone and has that cascading effect. So if we're increasing TRH, that means we're going to increase TSH. If we're increasing TSH, then we're increasing the release of thyroid hormone. So now our third step is to figure out where our error lies. And we're going to draw out some checks and X's. So we'll always start with the first hormone in our pathway. So if we have increased release of hormone 1, we should have increased release of hormone 2. So we'll give that a check. We have increased release of hormone 2, we should have increased release of hormone 3. So we'll also give that a check. We have increased release of hormone 3. Through negative feedback, we should have decreased release of hormone 1. So we see that is where our error lies, that negative feedback loop. The hypothalamus is not detecting that. So our error lies with hormone 1. So now we can compare our levels to the normalized data. So this first column and row is our normalized data. So we see that we have increased levels of TRH. So anything above 40, we have 85 and 55. We have increased release of TSH, so increased levels of TSH. So anything above 50, we have 75 and 82. And we also have increased release of thyroid hormones. So anything above 3, 7.5, and 6. So whenever we try and compare Colin to one of these data sets, we can say that he matches data set none. He matches none of these data sets. So Dr. Chang loves trick questions like that. He really wants to test your knowledge. And here is one of those trick questions where we see that Colin matches none of the data sets. And this would make sense because remember the condition we are given that he has is a rare hypersecreting hypothalamic tumor. So this is a rare condition, and in the hospital and clinical setting, they would not have generalized data for this. They would need to run more tests. We see that this is a condition that's occurring at that first integration center, the hypothalamus. So they would need to run brain scans, some MRIs, and do some much more advanced testing to figure out, what's going on here. So again, we have that negative feedback loop that's not working. So, we increase the release of TRH, we increase the release of TSH, we increase the release of thyroid hormone. So we're producing thyroid hormone, and through negative feedback loop, we should tell our nervous system, hey, we've released enough thyroid hormone. Let's slow down the release. However, our negative feedback loop is not working. Our hypothalamus is not detecting that negative feedback loop. So we keep releasing thyroid hormone. We keep releasing it, and our thyroid gland is growing, and we develop this big tumor of our thyroid gland because we just keep producing thyroid hormone, growing that gland, and forming tumors. So this is a rare condition that would definitely need further testing to determine how to treat. So Dr. Chang doesn't ask us to do this in these questions. He asks us to start doing this on question number 3. However, you can never get too much practice with questions like this. So we're going to add a little extra step to this. So what we're going to do is we're going to determine what type of pathology this is. So we have 2 types of pathologies. We have secondary pathology and we have primary pathology. So anything above this line is going to be a secondary pathology, and anything below the line is going to be a primary pathology. Now, I know that's confusing. You would think above the line, that's like primary, and below secondary. It's not that case. So how I remember that is there's 2 hormones above the line, hormone 1 and 2 are tropic hormones. That's a secondary pathology. Below the line, there's only 1 hormone, our final hormone. So that's a primary pathology. And pathology, what that means is some type of disease or condition. So something is malfunctioning. And we're going to get further into the definition of secondary and primary pathology. But for now, we're just going to practice determining which one it is. So in this case, what type of pathology would this be? It would be a secondary pathology because it's occurring in one of those tropic hormones. It is an error occurring above our line that we drew out. It's specifically occurring at the hypothalamus, so it would be a secondary pathology. All right, awesome. So let's move on to our next patient. So now we have our next patient in our room, and it's our job to determine where their error is occurring. So what we see is Sam has this iodine deficiency that's resulting in low production of thyroid hormone. So low production of thyroid hormone. So it's very important that we distinguish when we start drawing our arrows, whether it's affecting that final hormone or one of those tropic hormones. So where we start our arrow is very important. It doesn't matter if you start it with hormone 1 or hormone 2. If it's, if we're given some disorder that affects one of the tropic hormones, you can start with hormone 1 or hormone 2. However, if it's an error or condition affecting hormone 3, you need to make sure you draw your first arrow on hormone 3. So because we are told that we have low production of hormone 3, thyroid hormone, we will draw our arrow first at the thyroid hormone. And we are told that we are having low production. So we will draw a down arrow. We have decreased release of thyroid hormone. So we start there. So through negative feedback, we have decreased release of thyroid hormone. We will have increased release of thyroid releasing hormone. Thyroid releasing hormone is hormone 1, which has that cascading effect because it's a tropic hormone. And therefore we will increase the release of thyroid stimulating hormone. So now we've drawn out our arrows. Let's draw out our checks and X's. Let's figure out where our error lies. Whenever determining where our error lies, we can always begin with hormone 1. So if we have increased release of hormone 1, we should have increased release of hormone 2. So we'll give that a check. If we have increased release of hormone 2, we should have increased release of hormone 3. So there we go. That's where our error lies. To finish checking, if we have decreased release of hormone 3, we should have increased release of hormone 1. So we'll give that a check. So therefore, our error lies in the thyroid gland. We're having some type of disease or condition or pathology of the thyroid gland that's causing the incorrect release and production of thyroid hormone. Here we see it's a low production. So this would be considered hypothyroidism. So let's use our arrows to compare to our data. So we see we have increased release of TRH, so anything above 40. So we have 85 and 55. We have increased release of TSH, so anything above the normal level of 50, 75, and 82. And we have decreased release of thyroid hormone, or T4, so anything below 1, 0.05 and 0.4. So which data set does Sam match? He matches data set A. So data set A. So in the hospital and clinical setting, it probably wouldn't say data A. It would probably say hypothyroidism, which we know is the low production of thyroid hormone. This is a very common disorder. Many people have this. My mom has hyperthyroidism. My best friend has hypothyroidism. It's very common. It's not a life altering condition. It does not change your quality of life drastically. What it does is it affects your metabolism. So if you have hypothyroidism, you're gonna have slower metabolism, and you're not going to burn fats, sugars, things like that as quickly. So you won't burn calories as quickly, and it normally leads to weight gain. If you have hyperthyroidism, that means you have a very fast metabolism. So you burn calories much quicker, and you normally have a low body weight. So if you ever see those people that can just eat like so much, but are stick thin and never gain weight, they most likely have hyperthyroidism. And again, this is easily detectable, and it is fixed by just taking a medication, a pill that doesn't affect your quality of life that much. So now that we've answered the question, let's go ahead and add that extra step of determining which type of pathology this is. So we first draw out our line, and if it is above the line, it's secondary, and below the line, primary. So which type of pathology would this be? This would be a primary pathology, so a disease of the thyroid gland, hypothyroidism. Awesome. All right, so here's our last example, and we have a patient named Barbara. I'm actually going to pause this and animate it. I guess I forgot to animate this slide, so give me just a second. All right, I've now added in those animations so we can work through it together. So what we see with our last patient of the day, we have Barbara. Barbara has this autoimmune disease in which antibodies produced completely mimic TSH. So this one's a little complicated, but what we mean by mimicking TSH is we have decreased levels of T4, thyroid hormone. Why that is because if we're mimicking TSH, we're mimicking thyroid stimulating hormone. And the role of thyroid stimulating hormone is to stimulate the production of thyroid hormone. So we are stimulating the production of thyroid hormone. So we're stimulating the production or increase of thyroid hormone. So I know that's confusing. Anytime you see mimicking of, one of those tropic hormones, it means that we are causing the increased release of that final hormone. So in parentheses, next to mimic TSH, write increase T4. That's where we're going to start. So we draw our arrow first on T4 because we are stimulating the release of T4. These antibodies are mimicking the stimulation of T4. So our first arrow is the increase of T4. So if we increase hormone 3 or T4 through negative feedback, we will decrease the release of hormone 1, TRH. We decrease the release of TRH, then we decrease the release of TSH. So now we're going to do our checks and X's and figure out where our error lies. So if we have decreased release of hormone 1, we should have decreased release of hormone 2. So we give that a check. If we have decreased release of hormone 2, we should have decreased release of hormone 3 because hormone 2 is a tropic hormone and has that cascading effect. However, we see here it worked in the opposing direction. So our error lies with the release of hormone 3. So just to finish off checking, if we have increased release of hormone 3, we should have a decreased release of hormone 1 through negative feedback loop. So that is good. So here we see our error lies with the production of thyroid hormone. We are increasing, or we are overproducing thyroid hormone. So let's go ahead and compare this with our data. So we have decreased TRH, so anything below our normal level of 10, so 5 and 4. We have decreased TSH, so anything below our normal level of 5, 3 and 1. We have increased release of T4, so anything above our normal level of 3, we have 7.5 and 6. So which data set would Barbara match up with? She will match up with data set B. And in the hospital or clinical setting, I bet it would say Graves' disease instead of data B. So what clinicians and physicians and doctors and nurses can do with that is they can use that data, generalize the patient and treat them from there. Instead of having to run extra tests to figure out how to treat them, they have the data generalized. That data will line up with the specific treatment, and the doctor can prescribe whatever is needed to, fix the error that we found. So Graves' disease, the doctor would prescribe some type of treatment that would fix the increased production of T3 and T4 and bring it back to normal levels. So lastly, we're going to determine which type of pathology this is. So what do we do first? We draw our line. And if it's above the line, it's secondary, below the line, primary. So what type of pathology is this? This is a primary pathology. So a primary pathology. All right, nice. So our last question, we're using kind of a different method here, but it works just in the same way as our previous methods. Instead of being given a specific condition and being asked to compare it to data, we're just examining data itself. So this is how it would normally be done in a hospital or clinical setting. A patient would come in complaining of some type of issue. The doctor would run tests on them and their data would come up. And we would compare their data to these normal levels. So this is how it really is normally done in a hospital and clinical setting. So what we see is that for data set D, and again, here's those definitions for primary and secondary pathology. Primary pathology affects the final hormone in the pathway, so it affects that specific endocrine gland. And secondary pathology affects one of those tropic hormones, so either hormone 1 or 2, either the hypothalamus or the anterior pituitary gland. So again, we're looking at data set D. So we see that TRH for data set D is above normal levels. So we draw our up arrow. We see that TSH is below the normal levels at 1. So we draw a downward arrow. And we see that our T4 levels are also below our normal levels. So we draw a down arrow there. So now we do our checks and X's to determine what's going on. And so if hormone 1 is being increased, we're increasing the release of hormone 1, we should increase the release of hormone 2. However, that is not the case here. So there's where error lies. If you finish checking, you'll see that the other two have no errors and are working just fine. However, our issue lies there with the anterior pituitary. So if it lies with the anterior pituitary, with our second hormone, what type of pathology would that be? That would be secondary pathology. Secondary pathology. All right, great job, y'all. So definitely work through these a few times and make sure you can do them. You'll see something very similar to this on the exam. So make sure you work through these a few times and understand how to solve. All right, so let's move on to our next question. So this one should be really easy now because I've mentioned this many times. So we're talking about that hypothalamic control pathway again, specifically T4. So the release of thyroid hormone, which we just worked on. So what functions as the first integrating center? I really hope you'll have this down by now. That is the hypothalamus. What functions as the final integrating center whenever we're releasing T4? That is the thyroid gland. So if we were talking about cortisol, what would the final integrating center be? the adrenal cortex. If we were working with our sex hormones, what would the final integrating center be? It would be the gonads. In this course, the final integrating center for growth hormone would be that anterior pituitary, because we don't go as complex with that. All right, cool. So now we're asked, what is the first efferent? So whenever we say efferent or afferent, we're talking about the first hormone being sent or arriving. So anytime you see efferent, think efferent exit, E and E, efferent exit. Anytime you see afferent, think arrive, afferent arrive, A and A. So our first efferent hormone means our first hormone to leave one of our integration centers, the first one to leave the first integration center. So our first hormone to leave IC1 is TRH. So TRH. For cortisol, it would be CRH, cortico-releasing hormone. For GH, it would be growth hormone releasing hormone. And for sex hormones, it would be gonadotropin releasing hormone. Then we're asked, what is our final efferent? So our final hormone to exit the pathway. And that would be thyroid hormone. So that final one to exit. So I would definitely work through these with the other hormones and make sure you know the difference between the efferent hormones and the afferent hormones. So if we were to say, what is the first afferent? It would also be TRH. That's going to be the first one to arrive in the pathway, the first one to arrive at one of the integration centers. What's our second afferent? It's going to be TSH because it's the second one to arrive at the second integration center or third integration center, final. And then what's our final afferent? That's going to be thyroid. So our final afferent will be thyroid. So practice that a few times. You know what, actually final afferent is going to be TRH. So second afferent and final afferent are the same because we only use 2 integration centers in this course. We don't go into those middlemen. So that final afferent would be TRH because it is the last hormone arriving at our final integrating center. So I added some practice questions on the worksheet, so you can go ahead and practice those. And if you want the answers, either compare with each other or come to my Q&A on Sunday or my office hours on Monday. All right, so now we're moving on to the next question. So now we're kind of looking at the synthesis and release of thyroid hormone. So what we see is that thyroid hormone is synthesized or created inside of the cell. And what keeps it inside of the cell is this large protein called thyroglobin. So we synthesize and create thyroid hormone in the cell. And the reason it doesn't leave the cell is because it's attached to this large protein thyroglobin. So proteins trap hormones in a cell. Once we've synthesized and created that hormone, we want to release it from the cell so it can go to some area of the body and cause a response. So therefore, we will strip it from the protein. We will strip it from the thyroglobin, and therefore it can leave the cell. So let's work through the questions that were given. And now that we have this explanation, hopefully we can answer them. So by what method of movement do thyroid hormones leave the thyroid cell? If you remember back to discussion 1, steroid hormones, which is what thyroid hormone is, can move by simple diffusion. So our steroid hormones move by simple diffusion. T3 and T4 are steroid hormones. Therefore, they can simply diffuse through the cell membrane. We do not need to use a channel or carrier. We don't need to use the facilitated diffusion, which is channel or carrier. And we don't need to use active transport. So a symport, an antiport, or an ATPase subunit. They can simply diffuse past that cell membrane. Next question is, how does thyroid binding globulin, thyroid globulin, the large protein, function? Again, it traps T3, T4 inside the cell. So it traps that thyroid hormone in the cell while it's being synthesized and created. So you'll learn this in lecture if you haven't learned it already, but I wanted to include it here just to give you a second, drilling in of this information. So what we see is that our proteins are hydrophilic and lipophobic. So if they are hydrophilic, that means that they want to stay inside of the cell. And if they're lipophobic, that means that they are scared of the membrane. So hydrophilic, you can think they're kind of scared of the outside of the cell, and they prefer the inside of the cell. And lipophobic means they're scared of that membrane. Our membranes are made of fatty acid backbones. We have those two fatty acids that are attached. So lipo means fat, phobic means fear. So our proteins have this fear of crossing that membrane, so they stay inside of the cell. Compared to our steroid hormones, they are hydrophobic and lipophilic, so they want to leave the cell. They do not like the inside of the cell, and they want to get out of the inside of the cell. And they're lipophilic, so they can cross that membrane. They're not scared of the fatty acid backbone, and therefore they can cross that membrane through simple diffusion. So you'll go over that a little more in depth in lecture, but I just wanted to kind of reinstate that to y'all. So our last question here is give a teleological reason for why our thyroid hormones have a nuclear receptor. So if you remember back to the very first slide of our very first PowerPoint and lecture, teleological is talking about the why. In systems physiology, we have teleological and mechanical explanations. Mechanical gives you the how. Teleological gives you the why. So here we're asked, why do T3 and T4 have this nuclear receptor? And what we see is that steroid hormones, T3, T4, they have receptors that are on DNA itself. So on that DNA ladder. On that ladder, we have steroid hormone receptors on the nucleus. And those steroid hormone receptors act as transcription and translation factors. So if you remember back to like functional biology, our transcription and translation factors, their role is to take DNA, convert it into RNA, which will be converted into a single amino acid, which will form together with other amino acids to form a new protein. So steroid proteins, therefore, stimulate the production of new proteins. So they're on DNA and they act as that transcription and translational factor to convert that DNA into RNA, RNA into a single amino acid, which will be converted into a new protein. So therefore, we see steroid hormones stimulate the production of new proteins, and that's why they have that nuclear receptor. All right, awesome. So now we're asked which is more likely to be an effect of T3 and T4, so of our thyroid hormone. So either stimulate production of sodium potassium ATPase subunits or upregulate sodium channel activity by upregulation of IP3. And so the answer here is stimulate production of sodium potassium ATPase subunits. Sodium potassium ATP subunits are cell membrane proteins. So those ATPase subunits are cell membrane proteins, and we just discussed that steroid hormones' function is to produce new proteins. So our answer is stimulate the production of our sodium potassium ATPase subunits or our cell membrane proteins. If you're wondering what IP3 is and the second option, that deals with proteins, not steroid, or sorry, that deals with, let me see my explanation I had here. Sorry, I've taught this 3 times today. My brain is going numb right now. IP3 is a second messenger used by proteins, not steroid hormones. So we do not use that with our steroid hormones. They have no relation to IP3. Instead, we use it with proteins. And again, you'll hit that much harder in the lecture. But if you haven't heard it yet, that's a brief overview. All right, so now we're going to look at this graph, which looks really complicated, but I promise you it's not. What this is here is a radioimmunoassay. So it was like a blood smear that went through this process, most likely a spectrophotometer, and then graphed out in this computer system graph that we see here. So let's first figure out what this graph is telling us. So the x-axis is telling us the concentration of T4, and the y-axis is telling us the concentration of TSH. So we see T4 there and TSH there. So what that means is as we move along the x-axis, we're increasing the production of T4. And as we move along the y-axis, we're increasing the production of TSH. These middle values here, this big circle, this block of values, those are normal values. Here we see some pathological conditions. Those are what those values represent. So we are asked which symbol represents data from a hyperthyroid condition. So hyperthyroidism means you're increasing the release of thyroid hormone. You're releasing too much thyroid hormone and your thyroid is hyperactive. So that means we are moving along that x-axis with the increase of thyroid hormone. And if we look at the symbols that are represented by the movement along the x-axis, that would be the circles. So any data over here, if a patient did this radioimmunoassay and they have hyperthyroidism, they would match up with the data right here, those circles. Now we're asked, do the data indicate primary hyperthyroidism or secondary hyperthyroidism? So anytime we're trying to determine this, we can draw out our hormonal pathway for thyroid hormone, and we can work through our methods again. So we know we're increasing the release of thyroid hormone. I've talked so much today, forgive me. We are increasing the release of thyroid hormone if we have hyperthyroidism. So we start with T4 or T3. So if we have increased release of T3, T4, that means we'll have decreased release of TRH because that works through negative feedback loop. Decreased release of TRH means decreased release of TSH. So we draw our line, or sorry, first we do our arrows, or sorry. Let me take a quick pause. Forgive me, sorry, going brain dead, had to get some water and do some jumping jacks, get that blood flowing back to my brain. But yes, so let's go ahead and draw out our arrows here. So if we have decreased hormone 1, we should have decreased hormone 2, so we give that a check. If we have decreased hormone 2, we should have decreased hormone 3. However, here that's not the case, and therefore that's where error lies. To finalize our check in X, we will draw that negative feedback loop. We have increased hormone 3, we should have decreased hormone 1, so we give that a check. So which type of pathology would this be? We draw our line and we determine that it would be a primary pathology, specifically a primary hyperthyroidism. So it's primary hyperthyroidism. It is a disease affecting the thyroid gland, causing the increased production of thyroid hormone. All right, so second to last question here. We are asked to circle the correct reflex pattern by which the release of thyroid hormone occurs. So I want you to pause the video and kind of try and figure this one out. It's very simple. We've gone through this many times. S means stimulus, R means receptor, endo means endocrine gland. So go ahead and try and figure this out before I tell you the answer. All right, so our correct answer is C. We see that a stimuli arrives, it is detected by a receptor of the central nervous system, the hypothalamus. So we release a neurohormone from the hypothalamus and send it to our first endocrine gland, the anterior pituitary gland, which releases a hormone and sends it to our second endocrine organ, our specific endocrine organ, which will release a hormone which will travel to a target organ or tissue and cause some type of response. So that's another type of wording Dr. Chang can use on the exams. So be familiar with CNS representing the hypothalamus, endo 1 representing the anterior pituitary, and endo 2 representing our specific endocrine gland. All right, so this last question, Dr. Chang told us that we do not need to discuss this. It's asking for us to propose a mechanism by which thyroid hormone could be permissive to growth hormone. You will not be asked to propose any mechanisms on the exams or in this class. Instead, you'll need to identify these mechanisms. So I want you to review what you learned in lecture about permissiveness, synergism, and antagonism. So just review those three things and be able to identify them on the exam, and you'll be just fine. All right, so woohoo, we made it through Unit 1, discussion material. This should say officially a fourth of the way done with learning discussion materials because we have 4 units, but we just completed Unit 1, discussion materials. So great job, y'all. Be proud of yourself. Please don't view this material as hard and overwhelming and stressful. I did that last semester and it made it really hard to enjoy. This semester, I've changed my mindset and I view this as hard, but very interesting. It's very interesting stuff. This is what doctors and nurses and clinicians use to diagnose diseases in patients and treat patients. So this is stuff that the majority of the population does not know anything about. And it's really cool that we're learning this stuff. So try and think of this all in a positive light. And I promise you it's going to make learning it a lot more interesting and easier. So that's all I have for y'all. If you have not heard so far, I will be holding a Q&A on Sunday evening. I haven't decided the exact time, so probably around 6 or 7 p.m. And then I will have my regular office hours for questions on Monday from 2 to 3 p.m. Before our exam Tuesday. So if you have any questions over discussion material that you want to come ask me, I will be available for both of those. The Q&A will be held over Zoom and office hours will be held in person. And I will get to you with the location, but we will be in Jester for those office hours. Last announcement, we will have no discussion next week. You have your exam next week. So next Wednesday or whatever day you have discussion, take that day to either study or rest after your exam. So we won't have discussion next week. So I will see y'all for discussion number 4 in 2 weeks from now. And I look forward to it. Again, I have copies that I will bring with me to the classroom, but I only have 10 per section. So please make sure to print out your own copy. So anyone that, wasn't able to make it to a printer or something can use one of those extra copies, and it is first come, first serve. But yeah, so I will see y'all either in the exam if you're the 11 a.m. section, or I will see y'all in 2 weeks for discussion 4. Bye, everyone.